differences in gene expression that we observed in the Solanum lycopersicum homozygous plant were, as is described here in this picture, when we can observe that the plants carrying the penelase allele have 3 more higher levels of mRNA for BT3. Actually, when we differentiate the transcript coming from the different allele in the heterozygous, we found a picture like this. Coming back to the sequence analysis of these genes, what we found, as mentioned before, was these only differences in the protein, but more importantly, we found an insertion in the penelase allele and also an insertion in the lycopersicum allele. This insertion in the lycopersicum allele can be targeted, or at least this is the hypothesis that we have, targeted by a small RNA that is present in tomato small RNA databases. The hypothesis after this observation, after this result, was that the secondary small RNA generation can target both BT3 alleles. The model or the hypothesis, the model for this hypothesis is that in the case of MAT2 allele, this small RNA can target the mRNA of BT3, and the degradation of this mRNA can later target the penelase allele in the heterozygous plant, and this can explain the differences that we observe when we measure the mRNA for these genes. I think that I will stop here because I think we are running out of time, and I will skip this, and I would like to thank the whole group which are working, mainly to the people who have produced the result that I presented today, Gabriel Lichtenstein and Leandro Quadrana and Thomas Duffy, and also the partnership that we have with the laboratory of Magdalena Rossi at the Sao Paulo University and the laboratory of Alistair Fernie and Björn Useder at the Max Planck Institute, and also thanks the collaborator of my group, and also thanks the founding for different agencies that make possible our work at the Institute. Thank you very much. I will be happy to answer questions.
Okay, is there any question for Fernando? Okay, I have one. Are the GFP plants overexpressor plants, or yeah? Did you find any phenotype in plants or fruits? Nothing?
No, nothing. Actually, we didn't. Well, what we checked was actually whether there were some alteration in metabolism, because this was one of the points that we wanted to be sure about this plant, to use them as a model for the big experiment.
Yeah, exactly.
And we measured we did metabolic profile in the fruit and leaves of this plant, and we didn't find
anything,
substantial differences.
Okay. Okay, thanks.